Okay, so we spoke about passive transport, which is the type of transport that happens without us spending any energy. And now in this video, we'll have a quick chat about active transport, which is when we move stuff across the cell membrane for whatever reason, but it takes energy. We need to add energy in order for that process to happen. Now, a lot of times this will happen when we're trying to move objects from an area of low concentration to an area of high concentration. So if you think about diffusion, we're doing the opposite thing. In order for us to do that, we need to work against natural diffusion. So that's going to cost us energy. But active transport is a really, really important thing in our body systems because it will allow our cell to accumulate or to get stuff to concentrate inside of the cell. If you're a cell and you're carrying out thousands of metabolic reactions a day, it's important to have those materials on hand in order for those reactions to happen smoothly. So you don't want to wait until something can diffuse across the membrane all by itself. It doesn't cost any energy. What you want to do is, if that material or that substance, that reactant is present, you want to bring it into the cell and concentrate it in there so you've got a big stockpile and you can use it whenever you want to. And that's what active transport allows us to do. So it's absolutely necessary for our cells to survive. We have two general forms of active transport, and what they do is they allow us to actively transport things into the cell, okay, and we call that endocytosis. So endocytosis, if you look on the picture down there in the slide, is when we actively move stuff inside the cell. So the cell sees something it wants, and it uses energy to bring that inside the cell. The other type of active transport is exocytosis, and that's exiting the cell. So those two are pretty easy to remember. Endo means into, exo means out of. So in exocytosis, we have things inside the cell that we don't need, we don't want it there anymore, and we use energy to kick it out of the cell. Okay, so first let's have a look at exocytosis. Now, like I said in the last slide, this is how stuff exits the cell. So EX, exit, EXO, exit, cytosis, exocytosis. So it's an active transport, so we know it uses energy. And what happens is we get stuff inside of the cell, and the cell will form a vesicle around it. Now, a vesicle is a small organelle that occurs inside the cytoplasm. It's got a membrane around it, so it's a membrane-bound organelle. And we can put stuff inside of a vesicle. So a lot of times vesicles are used for moving things around or storage. Now in this case, the cell's got identified stuff that it doesn't want, and it has placed that in the vesicle. So if we start over here first, here's our vesicle, and that's full of stuff that we don't want. So what will happen is the membrane that's a part of that vesicle will approach the cell membrane, the plasma membrane on the outside of the cell, and that membrane will fuse with the cell's outside plasma membrane. And when it does that, the contents of the vesicle is free to leave the cell. And the fusing of that membrane is what costs us energy. So that's where we put the energy in. And then excuse me, the stuff that we don't want is kicked out of the cell. And you can see over here on the end, the cell has gotten rid of that material by exocytosis. So it cost us a little bit of energy, but we got rid of that stuff that the cell doesn't need anymore. Now, if we look at endocytosis, well, it's the exact same process, except it works in the opposite direction. The cell will identify something that it wants to take in. So again, we'll start on this side. The cell has identified this little pebble here that it wants to bring inside. So what it'll do is it'll form a pocket or an invagination of the cell membrane around that molecule or whatever substance it wants, and that membrane invagination, that pocket, will get bigger and bigger until the membrane here pinches, And what happens then is we get the formation of a new vesicle inside the cell with the substance that we wanted. And then we've got the vesicle is in there, and it's got whatever substance that the cell needed at the time. And then whenever the cell needs to use it, all that stuff is available inside that vesicle. Okay. So again, making that cell membrane fold and make a pocket around the substance that we want, and then joining the outer membrane together to form a new vesicle inside the cell, that's where we're going to use up the energy. So exocytosis, endocytosis, the process is the same, except it goes in the opposite direction, depending on if we want to get stuff into the cell or we want to get stuff out of the cell. Now, we're going to talk about two special cases of endocytosis here. So these are both going to be endocytosis. We're going to have phagocytosis and pinocytosis. So if you look at the pictures here, it's the same process that we just talked about in the last slide. The difference is what the cell is bringing inside. Okay, in phagocytosis, generally what the cell is bringing inside is bigger particles, chunks of substances that it needs, and in a way it would be a solid. Okay, a whole bunch of different things can be put in this. It could be amino acids, it could be proteins, it could be anything at all like that, but it's going to be a larger, more substantial molecule. When we talk about pinocytosis, what the cell is ingesting or what the cell is bringing inside is generally a liquid. Whoops, not liquid, liquid. So pinocytosis is when a cell engulfs a liquid, and phagocytosis is when a cell engulfs a solid. Sometimes you'll see them called cell eating and cell drinking. I don't like to use that because it makes it sound like the cell's got a mouth or a digestive system or anything like that. So I'll just generally call it phagocytosis when we're talking about a solid, and pinocytosis when we're talking about ingesting liquids. So here's a look at pinocytosis. We've got our solute here, which should be our solvent, I guess, if we're talking about pinocytosis. So the membrane folds in, it selects the objects that it wants. In this case, it's going to be some type of a liquid. The membrane folds around that liquid, and then the membrane pinches to become that vesicle. Once the vesicle is inside, the cell membrane is shut. It's closed again, so it maintains the protection and integrity of the cell. But we've got a vesicle, a membrane-bound vesicle inside the cell that the cell has raw material in it, and it can use for some type of reaction later down the road. Now, the last type of cell transport we're still talking about active transport. We're still talking about endocytosis, but now we're talking about receptor-mediated endocytosis. So this is a specific case, a very special case of endocytosis, and this involves a little bit more than just simply making a vesicle with the cell membrane. This involves special molecules on the outside of the cell that are used to trap molecules that the cell is looking for. So we call these, they're generally proteins, so we call them, they could be called a binding protein, or we can call them a receptor. And the receptors stick out on the outside of the cell, and they collect what it is that the cell is looking for. Now, another word for what the cell is looking for is a ligand, or a ligand, depending on how you pronounce it. So we have a receptor on the outside of the cell, and you can see in the picture here, we've got a receptor, and we've got that little green ball is the ligand. And once the ligand attaches to the receptor, then the normal part of endocytosis will happen. The cell membrane will fold in. and it'll start to form a pit around that receptor and the ligand, and then the ligand is brought inside in the form of a vesicle. Now, the receptors in this case can often be used again, and if you keep following that picture, this is a great example. When the vesicle is formed, it's called an endosome. So the endosome is formed. We've got our ligand inside, and then you can see another organelle here called a lysosome comes and it fuses with our endosome. So the ligand is inside there, and then the lysosome comes and fuses with it. Now, a way easier way to understand this is if you think about our immune cells. Okay, you think about our natural killer cells, our T cells in our immune system. Now, they have binding proteins on the outside, and they bind to specific ligands, but we call those antigens. Okay, and when those receptors, which are specific for, say, something like chickenpox, for example. So if you get exposed to chickenpox, these cells go around and they have receptors on the outside for the chickenpox virus. Those receptors will bind on to the chickenpox virus, and it'll start receptor-mediated endocytosis. So we'll have a whole bunch of those little virus particles that could cause chickenpox. They'll be stuck in the receptors, and the cell will form a endosome, so a pit or a vesicle inside with all of those little virus particles on there. Now, we want to keep using those receptors again, so we send another, once we make our endosome, we send another vesicle back to the cell surface so that the receptors for the chickenpox virus goes back on the outside of that cell so the cell can keep doing its job. Now, along comes this organelle called a lysosome. Now, inside a lysosome, we generally have all of these really nasty enzymes. Well, not nasty, but they do the hard work. Those enzymes will come in, and the lysosome will fuse with our endosome, so we get one big vesicle. And those enzymes that were in the lysosome can now attack and chop up and destroy those virus particles that cell captured. So receptor-mediated endocytosis is a really, really important process when we're talking about how certain parts of our immune system works.